Protein synthesis. Synthesis of a protein using the information stored in a gene is called gene expression. The two main steps of protein synthesis are transcription and translation. Transcription. Copying the relevant nucleotide sequence of DNA which codes the protein as an mRNA strand is called transcription. It takes place inside the nucleus. Transcription process consists of three steps. They are initiation, elongation, and termination. Initiation. The hydrogen bonds which exist between complementary bases of the two strands are broken by RNA polymerase, since RNA polymerase can perform the function of helicase. Elongation. RNA polymerase binds to the promoter region and starts the transcription process. It adds nucleotides and elongates the mRNA strand in 5' to 3' direction. Only one of the two strands is used as the template because it bears the promoter region which can bind the enzyme RNA polymerase. Termination. In prokaryotes, the transcription process is terminated when all the nucleotides have been copied. But in eukaryotes, the so-formed mRNA strand is called the pre-mRNA because it contains both introns and exons. Introns are the non-coding areas, while only the exons are the coding areas of a gene. Therefore, these non-coding areas are removed by a process called splicing. The mature mRNA formed after splicing leaves the nucleus and enters the cytosol. Translation. Conversion of the bases included in an mRNA strand into a series of amino acids is called translation. Translation process also consists of three steps, namely initiation, elongation, and termination. Initiation. mRNA strand binds with the small subunit of the ribosome. The large subunit also combines with them and forms the functional ribosome. Then the initiating tRNA molecule with the amino acid methionine binds to the A site, which is located in the large subunit of the ribosome. This functional ribosome with mRNA and the initiation tRNA is called the translation initiation complex. Then the mRNA strand moves as the codon AUG aligns with the anticodon of the initiation tRNA molecule, which is now at P site. Hydrogen bonds are made between AUG and the anticodon UAC of the tRNA molecule. This is the initiation of the translation process. Elongation. Then the second tRNA molecule with the relevant amino acid to the next codon of the mRNA strand reaches the A site. This process is called reading of codons and it occurs left to right. A peptide bond is made between the two amino acids located at A site and P site now. Translocation. The tRNA molecule which was at P site, that is the initiating tRNA molecule, leaves the ribosome via E site once the peptide bond is formed. Then the third tRNA molecule comes to A site with its amino acid. Then the second tRNA molecule, which is at A site, moves to P site. Once a peptide bond is made between two amino acids brought by second and third tRNA molecules, the second tRNA molecule also leaves the ribosome via E site. Termination. Elongation process occurs this way until a stop codon of mRNA strand reaches the A site of the ribosome. The process is terminated since no amino acid is brought by the tRNA molecule to A site. The completed polypeptide chain is released to the cytosol. Subunits of the ribosome and the remainder of the translation assemble fall apart.
How newly formed polypeptides are getting ready for their functions. Question number 1. Incorrect statement or statements about eukaryotic protein synthesis. A. Translation process of protein is started when tRNA with methionine reach to A site of the large subunit of ribosome. B. Termination of translation occurs when a stop codon reaches to P site of large subunit. C. rRNA stimulates peptide bond formation. D. Polypeptide produced at the end of translation is accepted as a functional protein. E. Energy for the translation process is provided by GTP. Question number 2. Incorrect statement about proteins. 1. Newly synthesized protein represents the primary structure of a protein. 2. A protein gets its functional form by folding and sometimes by post-translational modification. 3. Post-translational modification of a protein is taken place in polysomes. 4. Formation of glycoproteins, lipoproteins, and phosphorylation are examples for post-translational modification. 5. Some functional proteins such as insulin is made by cutting the single polypeptide at two places, removing the central piece and joining the two pieces. Question number 3. The selective degradation of proteins 1. Is an essential mechanism in regulation of cellular activities. 2. Occur in response to specific signals. 3. Occur mainly to degrade faulty or damaged proteins. 4. Takes place in the nucleus. 5. Help to rapid degrade of regulatory proteins. Question number 4. Incorrect statement or statements about myoglobin and hemoglobin. A. Both transport oxygen. B. Both are red pigments. C. Both carry 4 oxygen molecules. D. Both represent tertiary structures. E. Both bear ferrous ions. Question number 5. Incorrect statement or statements about RNA polymerase involved in elongation process of protein synthesis and DNA polymerase involved in elongation process of DNA replication. A. Both need primers to start the process. B. Both use any one of the two strands to copy new strands. C. Both can move only along 5' to 3' direction. D. RNA polymerase can perform helicase activity but DNA polymerase cannot. E. Both add nucleotides involving the elongation process.